Good evening, dear students. Welcome to my classes. I'm Dr. Iqbal Rashid Mir, and I will be teaching you the structure of DNA today. So this is the name of the chapter is Molecular Basis of Inheritance. Okay, and this is chapter number 4 in your 12th class. So in this chapter, we are going to study about DNA, search for genetic material, then we'll study the RNA world, then replication, transcription, genetic code, translation, and regulation of gene expression. Human Genome Project and DNA fingerprinting are also included in this chapter. Okay, so we will start the things first of all. We will study about the structure of DNA in today's class in detail. Okay, so what is DNA? Anyone? What is DNA? Deoxyribonucleic acid. This is the chemical name. Understood? Deoxyribonucleic acid. But many things, deoxy, ribo, then nucleic acid. What are these things? Yes, we are going to deduce everything. Don't worry. Okay. So first thing is deoxy and ribo. You know what is ribose? Remember the chemistry classes? Yes. See here. Ribose sugar. If we talk about it is 5-carbon sugar. Yeah, the structure is like this. Okay. So this is the structure of ribose sugar. If we see the chemical composition, so here we have OH. Again here on this position we have H and OH. On the third position we have H and OH. Okay, this is the third position. And fourth position we have H and CH2OH. Here we have the fourth and fifth position, and we have H CH2OH. Okay, this is the ribose sugar. Understood? So what is there? Oxygen. So students, you can see here. Okay, let me change the color of the pen. Okay, you can see this position, the second position. The second position, you can see OH is present here. Okay, that is why it is called as ribose sugar. Understood? So this is first position, second position, third position, fourth position, and fifth position. So this is the ribose sugar. If we are removing the oxygen group from this place, okay, then it becomes, yes, tell me, it becomes, I am writing only H here. Now this is deoxyribose now, okay, because oxygen has been removed from this place. So this is deoxyribose sugar now, okay, and in DNA, this sugar is present. Understood? So students, if we talk about the DNA, the search started from Mendel. Okay, he called factor, genes, etc. Okay, and they followed the transmission from generation to generation. They did not exactly knew that this is DNA or anything, but they knew that something is there that is responsible for the transmission of characters from parents to the offspring. Understood? So nucleic acid, we know it is of two types. It is deoxyribonucleic acid and ribonucleic acid. Understood? So nucleic acids, they can be divided into, just a minute, they can be divided to DNA, RNA. What is DNA? Deoxyribonucleic acid. And what is RNA? Ribonucleic acid. Ribonucleic acid. Okay. So in this chapter, we are going to discuss the structure of DNA, its replication, the process of making RNA from DNA, that is transcription, genetic code that determines the sequence of amino acid in the proteins that we will also study. Okay. So let us start with the DNA. So students, DNA is the genetic material in all the living organisms except some viruses where the genetic material is RNA. Okay. If we talk the number of nucleotides in the DNA in the E. coli, okay, they are almost 4.6 into 10 raised to power 6 base pairs. Okay. So DNA in the E. coli, just a minute. In E. coli, the base pairs, they are 4.6 into 10 raised to power 6 base pairs. Okay. And what about human? Humans, how many? Yes. Yeah. 3.3 into 10 raised to power 9 base pairs are present in case of the humans. Our DNA is linear. The bacterial DNA is circular. Yes, this is double-stranded DNA. This is also double-stranded DNA. And we know the DNA in the E. coli, it is present in the cytoplasm, and in the humans, it is in the nucleus. Understood? So what is structure of polynucleotide chain? DNA. So DNA has 3 components. Okay. So if we talk about the DNA, it has 3 components. So what are the 3 components? Let me check the color. So it has a nitrogenous base, it has a pentose sugar, and it has, what is the third thing that is present in the DNA? Phosphate group. Okay. So pentose sugar we know already. Yes. Let me change the color. We know the pentose sugar. Okay. This is the pentose sugar. It is the pentose sugar. Yes. Okay. You know, nitrogenous base. Okay. Nitrogenous base can be purine, it can be pyrimidine. So I will write here nitrogenous base. Okay, they are purines, they can be pyrimidines. Okay, so what are purines? They have double ring structure. They have 2 rings. Okay, so I will try to draw the ring of structure here. I will not draw the complete structure, just an overview. Understood? So you can see this is the first ring, and to it is attached the second ring. So these are the purines, and pyrimidines, they are just 1 ring structure like this. If we talk about the purines, how many purines are there? Tell me. Yes. Purines can be adenine and guanine. Okay. Adenine and guanine are 2 purines. And pyrimidines, they are how many? Cytosine, imine, and uracil. These are three pyrimidines: cytosine, thiamine, and uracil. These are three pyrimidines. Okay, so remember, thiamine, it is present only in DNA, and uracil is present only in RNA. Understood? So in the thiamine, what is the difference between thiamine and uracil? There is one difference between thiamine and uracil. In the thiamine, we have the methyl group on the 5th carbon position. It is not present in case of the uracil. So if we do demethylation of thiamine, you will get uracil. And if you add a methyl group in case of the uracil, you can get thiamine. The question can come like this in the exam. Understood? So these are nitrogenous bases. So nitrogenous bases are attached to pentose sugar at this position. You can see here. Just a minute. Okay, so you can see nitrogenous bases are present here at first position of pentose sugar. Okay, and the bond to which nitrogenous base is attached to the pentose sugar, this is called as N-glycosidic linkage. Okay, N-glycosidic linkage or N-glycosidic bond. Okay, so this bond that is present between the pentose sugar and nitrogenous base. Nitrogenous base can be purine, pyrimidine, we know. This is called as N-glycosidic linkage. Okay, on the other hand, on the 5th carbon, you can see here a phosphate group is attached to the pentose sugar. This phosphate group is attached to the pentose sugar with the help of which bond? Yes, everyone? Yes, this bond is called as, I will write it here, phosphodiester bond. Which bond? Phosphodiester bond. Okay. What is phosphodiester bond? Tell me. The bond between phosphate group and pentose sugar at the 5th carbon position. Understood? So what are the two kind of bonds? So we know DNA, complete DNA molecule, it has phosphate group, it has nitrogenous base, it has a pentose sugar, which is deoxyribose. These are the three components. So linkage between pentose sugar and phosphate bond, it is called as phosphodiester bond. The bond is phosphodiester bond. And here the bond between nitrogenous base and pentose sugar, it is N-glycosidic bond. Understood? Everything is clear? Yes, any doubt? Okay, now if we see the attachment of nucleotides, okay, nucleotides attach with each other and they form a polymer, okay, and it has a free phosphate moiety at the 5' end of the sugar, which is referred to as 5' end of the polynucleotide chain. Similarly, at the other end of the polymer, the sugar, it has free hydroxyl group at the 3rd position, which is referred to as 3' end of the polynucleotide chain. The backbone in a polynucleotide chain is formed due to sugar and phosphates. Okay, so I will show you the representation here. For example, this is the sugar. Okay, this is the sugar. Okay, here we have the 5' end. Just a minute, I will change the color here. You can see here we have the phosphate group. This is the 5th position. This is the 3rd position. Okay, so here also phosphate of group of previous sugar can be attached. Okay, so two sugars are linked in the DNA with the help of 5 and 3 bonds. So here you can see the phosphate group is present and this is linked to the previous sugar. So I will draw another molecule of sugar here. You can see here. Yeah, okay. Again, we have the 5. Here we have, sorry, this is the 3rd, this is the 5th, okay, and this is the phosphate Okay, and here we have the nitrogenous base. Here we have the nitrogenous base. So this is the second sugar. Similar thing happens also here. We have another phosphate. Okay, and this is attached to the next sugar like this. Yes. Okay, again another nitrogenous base and phosphate group attached to it. Okay, another molecule. Okay, let us draw. This is the third position again. This is the fifth position. So here we have another phosphate group. It is attached to the next nitrogenous base. Sorry, next pentose sugar. So again, we have the nitrogen atom here, the nitrogenous base. We have the third position, we have the fifth position here. Okay, so this is how different sugars, they are attached with each other through phosphodiester bond. And a polynucleotide change is found. Okay, so like these thousands of sugar uh, moieties, they are attached with each other through 3-5- bond. Okay, and this is 5' to 3'. The DNA, if we see the replication, it is also from 5' and to 3' end. They are nothing, they are the positions of sugar at which they are attached. So a polynucleotide chain is formed in this manner. Understood? Any doubt? So how the DNA structure was derived? It was derived on the basis of X-ray crystallography structure that was proposed by Wilkins and Rosalind Franklin. Another was Chargaff rules. So what were the rules of Chargaff? He proposed that purines and pyrimidines, they occur equal in number in the DNA. So let us suppose, let me show another slide. Okay, so Chargaff. Okay, he proposed certain rules. So he proposed that amount of purines, adenine plus guanine, they are always equal to amount of pyrimidines. What are pyrimidines? Cytosine plus thymine. Okay, they are 50% inside the cell. They are also 50% inside the cell. Overall, they are 100%. Total is 100%. Okay, so this is the first rule you have to remember. Second rule you need to remember is that adenine is always equal to thymine and guanine is also always equal to cytosine. So remember these two rules, you can solve any question. For example, if the question comes that inside a DNA, the cytosine is 24%. Okay, so you have to determine the percentage of adenine, guanine, and thymine. And you can answer it. Hurry up. So it is very simple to calculate. Okay, so cytosine is equal to 24%. If cytosine is 24%, guanine is also equal to 24%. Okay, so I will write here cytosine is equal to guanine is equal to 24%. So now what we have to determine? Adenine. Okay, adenine is equal to thymine is equal to 100 minus 24. What is 100 minus 24? Sorry, it will become 48 actually, 24 and 40, 24 and 24. Okay, so 100 minus 48, it will remain 52. 52%. Okay, so adenine is equal to thymine is equal to 52%. Okay, or it becomes 52 divided by 2, which becomes 26%. Okay, so adenine is equal to 26%, thymine is equal to 26%. Simple, isn't it? You can also answer like this. Guanine is equal to 24. Adenine and guanine, they are 50%. So minus 24 from the 15. It is 26%. When adenine is 26%, thymine is also equal to 26%. Okay. So these are the rules of Chargaff. Okay. Now we have the double helical structure of DNA. It was proposed by Watson and Crick, okay, in the year 1953. So there are certain salient features of double helical structure. It says that DNA consists of two polynucleotide chains. The backbone is constituted by sugar phosphate and the bases project inside. So you can see this is the backbone. Okay, the backbone is composed of sugar plus phosphate. Okay, and bases, they project inside. Bases hold two strands of DNA together. Understood? The two chains of DNA run in anti-parallel direction. Okay, you can see here anti-parallel polarity. This strand, the yellow one, it is 3' here, 5' on the other end. Okay, the green one, it is 5' here, it is 3' on the other hand. Okay, two strands of DNA are anti-parallel. Okay, if we see the length of one complete turn, it is 34 angstroms. Okay, or in other words, it is 3.4 nanometers. Okay, so 1 angstrom is equal to 0.1 nanometer. Remember. Okay, if we talk about the number of nucleotides in one turn, okay, number of nucleotides in one complete turn, they are 10.5. 10.5 nucleotides in one complete turn. And distance between two nucleotides, consecutive nucleotides, it is 0. Okay, it is 3.4 angstroms. Okay, so it is distance between two nucleotides, any two nucleotides. Another thing, DNA has a minor groove. Other groove, it is major groove. Major groove. Okay, the figure is from your NCERT book. Okay, you can see that the diameter of DNA, measure any region, the diameter, it is always 20 angstroms in diameter or 0.2 nanometer. Okay, oh sorry, 2.0 nanometer. Understood? So two strands are anti-parallel. The DNA is right-handed helix. Understood? So these were postulations by Watson and Crick. Okay, for their work, Watson and Crick received Nobel Prize in the year 1963, and they shared the Nobel Prize with Watson, Crick, and Wilkins. Fortunately, Rosalind Franklin died of cervical cancer. Okay, so students, this is all about DNA structure. Any question, any doubt? So with this, I'm ending the class. Take care, everyone. Goodbye. Take care.